All right, welcome to today's TED Talk on topic 9, which is all about transport, growth, and reproduction in plants. So you probably know a little something about plants coming into this. We know that plants have these different parts, these different organ systems, like roots that are in the ground, that are attached to stems, which hold up the leaves, and that in some cases in the angiosperms you'll have flowers. So all these different tissues are in existence in the plants. You also know that plants need sunlight to do photosynthesis and that they need water in order to survive. And that photosynthesis is occurring in the chlorophyll in the leaves in order to produce a sugar like glucose. And so we know what plants need. And so what we're going to focus on first is how they move these different materials around. So for instance, we know that water is coming from the ground and it's somehow being transported to the different parts of the plants. We also know that in photosynthesis, the sugars, right, are being created in these leaves during photosynthesis. And all the plant's cells are going to need that sugar, so there needs to be some mechanism of movement for those sugars that are also being produced. And so, just like humans have a vascular system in order to transport water and nutrients around our body, plants also have an analogous vascular tissue system to also transport their water and their sugar to the different cells in the plant tissue. So that vascular tissue has special names, xylem and phloem. And so these are going to move two different types of materials. So xylem is what is moving both water and minerals from the roots up throughout the plant. And we'll talk more about this process that we call this is transpiration, right? So xylem moves the water and minerals from the roots to the leaves through a process called transpiration, right? And so phloem, on the other hand, right, because there are these two tissues, phloem moves the food. Phloem is going to move a sugar that's produced in the leaves to the other parts of the plant, right? So sunlight is being used to do photosynthesis, which is creating sugar, and that sugar is being transported to various parts of the plant via the phloem, and that process is translocation. So one more time, xylem transports water and minerals from the roots to the leaves to the rest of the plant via transpiration, and phloem transports sugars or organic compounds. That's the other way that Ivy likes to say this. So phloem transports those organic compounds from where they're being synthesized in the leaves via translocation. And that can go in either direction. So when we're looking at the structure of xylem and phloem, not only do they transport different materials, but structurally they're very different. So whereas xylem is mostly hollow, rigid, dead tissue, phloem, on the other hand, has a lot more components. These are going to look different in cross sections. So where xylem is hollow, rigid, dead tissue, phloem is living tissue with a lot more components. So phloem has three different parts. It's got these sieve elements here, which have sieve plates on either end. And then you've got companion cells. These are little friends. They're like helper cells that are sitting just adjacent to the phloem itself. And those companion cells are transporting sugars, those organic compounds, via something called plasmodesmata. So these are the openings between the sieve element and the companion cell through which something is being transported. So just like one more time. Xylem, rigid and hollow dead tissue. This is transporting water via transpiration, whereas phloem is now living tissue with sieve elements that come in three parts. Companion cells on the right side, sieve plates on the end, and plasmodesmata, which are the openings between the companion cells and the phloem itself. So when we compare and contrast these two, xylem moves water, phloem moves the food. Xylem is dead tissue, whereas phloem is living. You've got these companion cells transporting an organic compound we'll later learn is sucrose through these plasmodesmata. So you might see longitudinal photos of these different tissues. And so if I took a stem here and I was cutting sections from the top down, so like lengthwise, you would get a structure that looks like this. And in this case, the xylem is going to have a lot of like stripy kind of marks to it, whereas the phloem is going to be nearby and it's going to look more like little bone structures. So even though the xylem is hollow, when you cut it open longitudinally, it's going to look like you have a lot of little lines across it, whereas the phloem is going to appear more empty. So you may also see cross sections, right, of the xylem of the phloem. And I have this video just so we're clear on how these are being created. You're going to like take a stem and you cut off like the top part of it in like little slices. So that's what a cross section is going to be, right? And so you can take cross sections of different tissues. And so if I take a cross section of a stem, this is what that is going to look like in a dicot plant. And we can see the vascular structures in here. So these little areas here, right, these little sort of acorn looking things, these are vascular bundles. And just a reminder, right, that xylem and phloem are both vascular tissues. It's just that xylem moves water via transpiration, whereas phloem moves them organic compounds via translocation. So both those vascular bundles are going to be bundled together. And so if we look at these, right, so this itself is a vascular bundle, so is this here, so is this there, so are these all. These are all vascular bundles. But in here, if I look between these two tissues, right, one of them appears to be like more hollow, right? So in this case, the xylem is kind of like hollow, which fits with what we know about its structure, that it's mainly hollow tissue. So when I'm looking at these cross sections, these little areas of hollowness, those are going to be the xylem. Whereas like the top of the acorn, the one that's a little bit more crowded, that's going to be the phloem. And so this is what they look like in stems. So if I take a cross section of a root, in this case, X marks the spot, right? So this X structure, this really hollow structure, is going to be xylem. So there are large kind of openings. And then right next to them in these grooves here, that's where the phloem is going to be. So phloem is here, phloem is here, but the X part is going to be the xylem itself, right? And so if I examine, this stem, I know it's a stem, right, because it doesn't have an X on it. And I would ask you to find a vascular bundle. I would pick some sort of acorn looking thing here, so that would be a vascular bundle. And if I then asked you to identify this structure, you would know that that's the phloem, versus these sort of larger bubbles are going to be the xylem, right? And so yeah, hollow xylem, acorn looking things, tops phloem. So in the roots, just a reminder that X marks the spot. So this is going to be the xylem, whereas these regions in here are going to be the phloem. Okay, so be ready to identify those. So going back to transpiration, right? Going back to the xylem, we know that xylem is moving the water and minerals. We know that the water is traveling from the roots up. And so in order to understand how that's occurring, right? So this process is called transpiration, the movement of water from the roots up through the plants and out through the underside of the leaves. So this process is called transpiration. And the reason that it works is because we're making use of the adhesive properties of water, right? And so back in 2.2, we talked about adhesive properties, cohesion and adhesion. So just a reminder that cohesion is water bonding to itself, right? So water likes to make little chains. If I pull on one chain, the rest of the water is going to follow with it, right? Versus adhesion is a tendency of water that's going to want to stick to another polar substance. So you know that if you stick your hand in water, some of that water is going to stick to you. So that's water adhering to you. So both of these adhesive properties are why transpiration works, right? So because you're getting some capillary action happening, and you're also including transpiration, which is going to pull a chain of water behind it, right? So just by itself, water is going to adhere to different parts of the xylem, right? Because water is a polar molecule, so it's going to adhere to the xylem. We know that water is going to have cohesion between other water molecules, and so if I pull a chain of water, through the underside of these leaves, that's going to give me something called transpiration pool. And this will pull up the column of water directly behind, right, from the roots through the stem all the way through the underside of the leaves. So how we do this, right? I'm showing water exiting the underside of the leaves, and how they exit is through a structure called the stomata. So we've looked at leaves. You probably look at a leaf and you don't think that much of it. You're like, oh, it's very simple. It's just this flat thing. But actually, there's quite a bit going on with the leaf itself, right? And so if we look at the leaf structure, this flat, simple thing is actually kind of complicated, right? So we have a cuticle on the upper epidermis, which is helping to, keep water inside the plant. And then you've got the presence of these vascular bundles, right? The xylem and the phloem. So this is transporting water from the roots up to the leaves. And you've got the phloem, right? So in here are also mesophyll cells. This is where most of photosynthesis is occurring. So that sugar that these mesophyll cells are producing are going to go into the phloem, right, to go to the rest of the plant. So leaf has a cuticle. It's got mesophyll cells, which are doing the actual photosynthesis. Xylem is bringing in the water to provide that water for photolysis in photosynthesis. And once the sugars are created by those mesophyll cells, those sugars are then being kicked back out into the phloem through these vascular bundles. So even if you look like a regular leaf, it's got these veins on it. Well, that's the vascular bundles of xylem and phloem that are moving around the leaf. So going back to transpiration, right, we have a top side of a leaf and we have an underside of the leaf. And this underside of the leaf has the stomata. And so these stomata, and they look like really weird mouths, is where gas exchange is occurring within the leaf. So if you look at the word transpiration, it sounds a lot like respiration, right? Or inspiration. It sounds like breathing, because functionally that's what transpiration is. It's the breathing of the plant. So that breathing happens at the site of the stomata. So these stomata look like little mouths. They can open and close because that's where the gas exchange is coming out. So when they're open, carbon dioxide gas is going in because I need that CO2 for photosynthesis. And at the same time, oxygen is coming out, right? Because that's a waste product of photosynthesis. So this is where gas exchange is happening. And also here is where the water vapor is being released. And so this is really how transpiration is working. That as we, right, let me get rid of this. Erase all ink. As we are pulling up water, right? So water is coming up from the roots through the stem. The stomata are going to open on the underside of those leaves and then basically exhale out water vapor. And as that water vapor is exhaled out, it is pulling up the chain of water behind it via transpiration pool. And that's how transpiration is actually working, right? As through the underside of those stomata. So stomata can either be open, right, or they can be closed. They have a choice whether to be open or closed. And so you want to be open when you're doing photosynthesis, right? Because when I'm doing photosynthesis is when I would need carbon dioxide, and is when I'm creating oxygen as waste gas. So the guard cells on either side are going to cause the stomata to be open in those instances. So during the daytime, right, the stomata are open because I'm actively doing photosynthesis. I need to have gas exchange. However, they can be closed, right? And so these will occur, the closed state will occur during nighttime, as an example, right? Because during the nighttime, I'm not doing photosynthesis. I have no cause to have gas exchange, so the stomata will close. There are other instances, right? Ooh, get away from that. There are other instances. Well, that got weird. I'm waiting for the. There are other instances where I would want to close this stomata. So for example, right, when the stomata are open, I am losing water. And so if water becomes scarce, then I need to close these, right? I need to close the stomata to keep myself from losing water. So there are appropriate times and places for those two respective things to be happening. Okay, so transpiration can be measured with something called a potometer. So this is not the best way to measure transpiration, but it is a way. So how a potometer works is you take a plant and you expose the leaves to the air, and you have the stem or the roots attached to a tube filled with water. So what should happen, right, is that as you have transpiration that is occurring when the stomata are opening, you're going to get transpiration pool on the water that is attached to this tube. And as that pool occurs, this water is then going to move, and you can measure the movement of that water using a ruler that is next to that tube. So this entire setup is called a potometer. So as that water is evaporating from the underside of those leaves via transpiration, that water vapor going through the stomata, that transpiration is going to pull the chain of water and move that water column. And again, you can measure that with a ruler. To reset a potometer, you have something called a reservoir and a tap. So the reservoir is going to store new water to help reset the potometer, or to allow it to resupply water should you run out, right? So transpiration is measured with a potometer. So there are some factors that can affect the rate of transpiration, right? So if you think like fundamentally, this process is about a couple of things, right? It's about how fast can evaporation occur, and it's about what factors will make that evaporation either faster or slower, or what are some instances in which a plant might want to hold on to the water that it has, right? So when we're looking at potometer experiments, there are a couple of things that can alter how quickly or how slowly that happens. And so some things, I'm just going to list them for you, right, are things like temperature and sunlight. So both of these are going to increase the rates of transpiration. The only one that decreases it is humidity. We'll talk about why that might be. And the one that is strange is wind. So initially, right, if I use a blow dryer to dry my hair, it is definitely going to increase the rates of respiration. So if you're a plant, you are going to lose water if it's windy, but maybe if that wind increases too much, the rates of transpiration are actually going to decrease. And so one of your tasks are going to be to identify the graphs of these different factors affecting the rate of transpiration. So if we look at temperature, right, when it's cold, you're not going to have a lot of evaporation of the water, so your transpiration rates are going to be pretty low. Compared to as the temperature increases, your rates of respiration are going to increase, right? If it's hot outside, you're going to have evaporation of water quickly. So light intensity is similar, that when there is low light intensity, right? So not only do you have low evaporation rates, but probably your stomata are closed under low light conditions. And as light increases, as temperature increases, right, those stomata are going to become open until you hit some sort of maximum, right? And so in this instance, the stomata, they're open, but this looks like one of the graphs that you saw in 2.5, right? The one of substrate concentration, where under low light, because light is a substrate of photosynthesis, so under low light, there's not much going through photosystems 2 and 1, and so you don't have a lot of enzymatic activity, but as the light increases, it will plateau once you have maximized, your enzyme usage. So photosystem 2 and 1 can only process so much light. Any more than that is not going to add any more product. So the similar way, you're after a certain amount of light intensity, you're not going to make transpiration any faster. So both temperature and light intensity, for the most part, are going to increase the rates of transpiration, right? So humidity, on the other hand, is the opposite. So if you think about if you're super sweaty and it's humid outside, sweating isn't really going to help you that much because there's nowhere for that water vapor to go. So for plants, it's the same. If they're releasing water vapor into the air and the air is already saturated with water, if it's humid, there's not going to be really anywhere for that water and transpiration to go, so transpiration rates are really low under high humidity conditions. So compare that to when the air is very dry, right? If the air is very dry, we know water is going to evaporate really quickly, so you're going to have really high rates of transpiration, right? So that water vapor is going to leave those leaves very quickly versus not so much when it's humid. So wind speed, right, starts low. At low wind speeds, you don't have much transpiration. You're not helping it evaporate that much, but as you increase the rates, you are going to hit some sort of a peak. And so you know that if you have some water on your desk and you blow on it, it'll dry faster if you're kind of blowing air across it. And so a plant is going to lose water under those low wind conditions versus if it's in a monsoon, it's probably going to close itself up because it's just trying to save itself. So just to recap, temperature and light intensity are generally both going to increase rates of transpiration, whereas humidity is generally going to decrease rates of transpiration, and wind speed is the one that kind of flips, where it's initially increasing to a certain point and then will start to decrease rapidly. And so you might be asked, say I'm doing a potometer experiment, right? And so remember, potometer experiments are what is allowing me to measure rates of transpiration, and say I'm studying temperature. So if I want to study temperature, then the question would become, what other factors do I need to control if I only want to study temperature? And well, it's basically, if I'm studying temperature, everything else needs to be controlled. If I'm studying temperature, humidity needs to be controlled, wind speed needs to be controlled, light intensity needs to be controlled, because any of those could alter the results of that potometer experiment. And it works the same for every condition. Say I wanted to study humidity, the effect of humidity on transpiration. Well, okay, then I need to control temperature, wind speed, and light intensity, because again, any of those could affect it as well. So same with light intensity. If I'm studying the effect of light intensity on transpiration, all these other factors would need to be controlled. Okay, so just to recap, xylem is moving water and minerals from the roots up to the rest of the plant via transpiration, and I measure the rates of transpiration with a potometer. And so that's compared to phloem, which is following the movement of the food, and that is called translocation. And just a reminder, xylem, hollow, mostly rigid dead tissue, whereas phloem is living. And we have these three components: the sieve element with sieve plates on either side, the companion cells, which are there to provide ATP and are really driving the transport of those organic compounds, and the plasmodesmata. So phloem, sieve element, sieve plate, companion cells, openings are plasmodesmata. So phloem is moving the food, right? And we're calling that process translocation. I'm moving locations. And so this is also referred to as the transport of organic compounds. And so what we're doing is we're moving those organic compounds, or specifically sucrose, from where it is being produced, the source, which is the leaves, those mesophyll cells in the leaves, and it's being translocated to different parts of the plant, known as the sink. And so sinks are areas of rapid growth, and if you're a plant, you've got two of them. You grow down in the roots, but you also grow up in the shoot. So the source is the leaves, and the sink can either be the roots or the apical shoot where the plant is growing upwards. Okay, and we're moving that food through the phloem, and that process is called translocation. So, what is interesting is that sugar that is being moved around is not glucose. It's actually sucrose, which you know as table sugar. And probably the reason that we eat sucrose as table sugar is because this is the sugar that plants are being, that plants are transporting all over their stems. So if you ever watch people who grow sugarcane, they can cut that sugarcane open and then chew on it and get sugar because there's sugar being transported, specifically sucrose, being transported through that plant. So it's not glucose that's being transported during translocation in the phloem, it's sucrose, which is our disaccharide, right, of glucose and fructose. So what is happening is that photosynthesis is creating glucose, and within that mesophyll cell, within that source cell, it is further creating the disaccharide of sucrose. That sucrose, right, is then going to be actively transported into the companion cells. And from here, that sucrose is then going to be actively transported through the plasmodesmata into the phloem itself. So step one, I make sucrose in my source cell. That sucrose is actively transported to the companion cell, and then that sucrose is further actively transported from the companion cell across the plasmodesmata to get into the phloem. And so you'll also notice that right next to the phloem is the xylem, which we know transports water. We also know that water crosses membranes via osmosis, and that when there is a high concentration of water near an area where there's a low concentration of water, that water is going to move passively from an area of high concentration in the xylem to an area of low concentration in the phloem. So again, that's the process of osmosis, and this is going to happen passively. So what that's going to do once that water comes in here is create an area of high pressure. And that sap that I've now created is not going to want to stay there. It's going to want to move, right? It's under pressure, so it will move from a source area to some kind of sink. So once it reaches an area of a sink cell where there's a companion cell, that companion cell is now going to, again, take the sucrose now out of the phloem into itself, and then actively transport that sucrose into a sink cell. So that's the destination, either in the roots or the shoots. And so now once we're here at the sink, the water situation has flipped. I have high concentration water in the phloem and low concentration in the xylem, and that water will once again travel via osmosis back to the xylem, where it's going to be transpired up the rest of the plant, right, via transpiration in that xylem, and will again keep crossing over via osmosis into the phloem and restart the whole process of translocation, right? So these are the steps of translocation. Source cell produces sucrose, actively transports that sucrose to the companion cell, which actively transports that sucrose through the plasmodesmata into the phloem. Water will then diffuse via osmosis from the xylem, create high pressure in the phloem, and force that sap to move from a source area to a sink area. Once it's in the sink area, the companion cell will then further remove that organic compound, that sucrose, into itself, and then further transport it to a sink cell. And then the last step is water moving back from the phloem to the xylem via osmosis, and the water is further transported, or transported up the stem via transpiration, and the cycle starts again. So just like we had potometers to measure transpiration in a plant, there is a system to measure translocation in the plant, and it's a little bit insane. So we measure rates of translocation via something called an aphid stylectomy. And so you're going to take a plant You lay it kind of on its side, and then you stick a bag over its head. And in that bag, you place radio-labeled carbon dioxide, right? And so plants are taking in that carbon dioxide. Rubisco is going to fix that into glucose, which will become a part of sucrose. And a sane person would have put a Geiger counter somewhere over here to track the movement of that carbon dioxide, but of course that's not what they did. Instead, they basically got a bug, right? And they put the bug down here, the bug's being an aphid. So as that CO2 becomes fixed, it's going to be radioactive. It's going to be radio labeled. And that CO2 is going to find its way into sucrose, which is now also radio labeled. And as it's translocated down the plant, it's going to travel with that radio label. So how they track the rates is they place an aphid on one part of the stem, and that aphid has like a beak, like which is called a stylet. And so it uses that stylet to feed on the sap. And then they basically record how long it takes the waste product of that aphid to become radioactive. And so that's how they measure the rates of translocation. And so this whole process again is called an aphid stylectomy. So we measure transpiration with a potometer, we measure translocation with an aphid stylectomy that involves an aphid and radio-labeled carbon dioxide. Okay. So, as we know, plants start from little seeds and they grow in two main directions, up towards the sun in the shoots and down towards the ground in the roots. And so this tissue here on either end has a special name, and we call them meristems, not because they're exactly happy, but these are basically like undifferentiated tissue in the plant. They can become many things. And so for a plant to go from a seed to a full plant, it needs to generate lots of new cells. So mitosis is going to be very active in a growing plant. And just a heads up that just like we have hormones, plants also have hormones, and their main hormone is a hormone called auxin, which is really going to promote that cell growth, and it also promotes cell elongation. So auxin, two jobs, promoting cell growth and promoting cell elongation. So when we look at the growth, right, in a plant, we've got two areas of growth, and they're both called apical meristems. You have the apical meristem in the roots, and you have the apical meristem in the shoots. What is important to note about these is that the cells are called indeterminate, which is just another word for undifferentiated, which is just another fancy way of saying that the cells that are in here don't exactly know what they're going to be when they grow up. They can still be anything. So in this apical meristem, right, we're here in the actual stem, the cells here can become anything that's in the shoot of a plant. They can become a stem, they can become xylem, they can become a phloem, they can become leaves, they can become flowers. So indeterminate means it can become any of those things. And this is definitely a high area of active mitosis because you're growing in that direction and making more cells. So in the meristem, you have lots of mitosis, and they are indeterminate. They can become many different cell types. And so as a plant is growing, right, it can decide to grow towards things that it wants. So for example, light, right? So phototropism is the response of plants growing towards light. And that is governed by, again, that hormone auxin, right? Auxin, main jobs, promoting cell growth and elongating the cells. So if I look at a plant that is undergoing phototropism, it is going to bend toward the light. And part of what is driving that is the expression of that hormone auxin. So auxin is going to be expressed on the shaded side, not the light side, the shaded side. And on that shaded side is where the cells are going to elongate, and they're going to replicate to allow longer curvature on this side to allow it to bend. So gravitropism works pretty much the same way, right? Plants can tell up and down, and part of how they do this bending is again through the expression of auxin. So on that curved side, that's where you're going to have the expression of auxin because it's elongating the cells, it's replicating cells to allow that bending to occur. And in both cases, auxin is passed between different cells via these efflux pumps. So to get it from the light side to the shaded side, I push it using these efflux pumps. And so in that way, auxin is able to tell a plant, okay, on this side, you're going to elongate and you're going to replicate so I can create a bend here. So that's how tropisms are governed. So our last topic is reproduction, right? And so we know from our plant phyla that angiosperms were the flowering plant, that these were hugely successful plants in the plant world, and that they have pretty complex structures, right? And so that flowers have both male and female parts. But let's start with like the easy stuff as far as labeling the flower. So if you look at a flower, you know that the pretty parts are called the petals, right? And then they have these like leafy green things, the sepals underneath that kind of protect the flower before it buds. So as mentioned, right, flowers have girl parts and boy parts both. The girl parts are called the pistil or the carpel, and it has really 4 parts. The stigma, which is sticky and catches the pollen. The style, which is attached to the stigma, it's helping to hold it up. And then you have the ovary, and contained within the ovary are ovules. So don't forget about the ovules in here. So stigma, style, ovary, ovules in the center. The whole part is the pistil and the carpel. The male parts. The entire thing is called the stamen, and it's got 2 main parts, the filament, which is holding up the anther, right? And so the anther is the pollen, which is basically fallible sperm. So this is the location of the male gametes, whereas in the female parts, that was the ovules in the ovary. So male parts, stamen, anther, which is where the pollen is, and filament, and female parts, stigma, style, ovary, and then ovules. So, when a mommy and a daddy plant love very much, right, what's going to happen is they are going to want to pollinate each other, right? And, oh, sorry, let's back up for a second. So the making of a flower, right? A plant has to decide very carefully when it is going to flower. And if you think about the season that's best for it, it's probably the springtime, right? And so in the springtime, conditions are favorable. It's warmer, right? So temperature is going to affect when these plants flower. And it's also following the light, right? Because there is generally more light in spring than there is in the winter or the fall. So how plants to decide to flower or not is by following either the amount of darkness or the amount of light in any given day. So there's 2 different ways they can do this. And so because of those 2 different approaches, you can either have a short day plant or a long day plant. And this is how they're going to decide when to flower. So we call that photoperiodism, is how it decides whether to flower, and it comes in that short day versus long day plant. A short day plant is following amounts of darkness. So it needs a set amount of darkness before it will flower. And so probably these are the plants that are going to want to flower during early spring, right? So I have these different amounts of daylight, and for a short day plant, I'm following amounts of darkness. There's some critical level of darkness that I will not flower unless that critical darkness period is exceeded. So if I don't have that, right? Say I have too much sunshine, no flowering, because a short day plant needs longer periods of dark. If I exceed, right, that dark period, then I am going to have flowering. But if I interrupt it, I will also have no flowering. So short day plants need uninterrupted darkness in order to flower. So long day plants are the opposite. They're looking for a critical minimum of having light around. So in this case, once I've exceeded the minimum amount of light, then I am going to flower. But if I have too much darkness, I'm not going to flower. Or if I interrupt my light period with any kind of darkness, then I'm still not going to flower, right? So short day plants minimum amount of darkness. Long day plants, minimum amount of light. So how that is tracked by a plant is not through the chlorophyll that's in photosystems 2 and 1 during photosynthesis. It uses an entirely different chemical called phytochrome. So phytochrome is a chemical that switches between an inactive state, noted by PR, so PR is inactive, and it can be converted to PFR if it is active. So red light, right, is a big component of daylight. So phytochrome can move from its inactive PR state to its active PFR. Whereas if there is more far red light, it will become inactive. And so that is how a plant is tracking how much light, right, how much daylight there is to decide when to help it flower. And so we know that we can hop down to HEB and buy flowers anytime we want. And those farmers are not sitting around waiting for that flowering to happen. They control, right, the light that is being exposed to the plants under their care. And so they can force those short day plants out to flower out of season by something called flower forcing. And so if I'm a short day plant, I just put it in dark, right? Longer than I normally, or long enough to induce that flowering. So that's called flower forcing. And so once I have a flower, right? Okay, mommy and daddy flower love each other very much. The daddy flower is producing sperm via pollen on that anther. It will eventually fly around and get caught by a sticky stigma, right? So say a pollen grain from another plant lands on that sticky stigma. It's going to create a pollen tube to go down the style. And so once those gametes are there, they're going to fertilize the ovules that are in the ovary. And at that point, a seed or a fruit can be made, right? So not to like ruin your day or anything, but seeds are basically plant embryos. These are little baby plants, and you eat and murder them all the time. So one of our tasks is to draw, right? Draw and label a seed. And so a seed will have, I'm going to just do this here. A seed is going to have an outer coat, and this is either referred to as a seed coat or as a testa. I've seen it referred to that quite a bit. So when you're drawing these on your own, outer layer is called the testa. The baby plant, as it's forming, has a number of different parts. The radicle, if you cut beans open, you can see them, right? The radicle is going to be the root, and it is on this side right here. The epicotyl is going to become your first leaves versus the hypocotyl, which is going to probably become the stem. And then what is left over, right, is this open space called the cotyledon. And so if you think about, right, baby plants, these plant embryos, they start life underground. And so they're not going to be doing any kind of photosynthesis. But because it's growing, it definitely needs access to a large amount of energy. And that energy is stored in this open space, in the cotyledon itself. So that's what we call this open space. You love eating beans, which, by the way, are baby plants, you murderers. You love eating beans because this is full of starch in the cotyledon. So this starch is here to support the initial growth of the plant embryo. So just to recap, when you're drawing a seed, outside is seed coat. I prefer that you use the word testa. Radicle in this corner is going to become the root. It's attached to the hypocotyl, which is attached to the epicotyl. So these are the first leaves. And then the rest of the space is the cotyledon, which is full of starch. So if you think about the requirements, what does a seed need to actually germinate? And it's different than what you'd think. So for sure, you need water, right? I can germinate a bean in like paper towels that are soaked in water. So you need to have water in order for germination to occur. And so if you think of your like a plant embryo, right? You're sitting out there in the dirt. You want to grow when it's the rainy season, but you also want to grow when it's warm. So temperature, right? Ideal temperature and water availability are going to be required for germination. And then the last thing that kind of surprised me was that you need an oxygen supply. So remember, the plants are full of mitochondria, just like you and me are. And those mitochondria, right, because this guy is growing and it's going to need to burn all the starch that it's here in the cotyledon, and it burns that starch in the mitochondria, right, to create energy, which is going to fuel its growth. So it does need oxygen in order to germinate. So key requirements in order for a seed to germinate: water availability, appropriate temperatures for growth, and then finally oxygen supply. So that's going to close out our topic 9.